Hello to everyone joining us and welcome to SensoGen's webcast. My name is Tina Lawson. I'm managing marketing and communications at SensoGen and I will be your host today. Today's talk is about the ESAC opinion on GARD that was released a few days ago, and we will start with a 45-minute presentation followed by a Q&A session. Today's presenters are CEO Axel Sjöblad and Chief Scientist Henrik Johansson. A couple of notes before we start. This webcast will be recorded and the recording will be available also on the investor pages on our website later on today. And regarding the Q&A, you can submit your questions at any time during the presentations. Just write your question in the questions chat box in the GoToWebinar panel. This chat box is located on the right side of your screen. You should be able to find it there. So just use it anytime you want to ask a question. So by saying that, we're now ready to start the webinar, and Axel's presentation will be the first one out and Henrik's will follow. So next slide, please. And Axel, please go ahead.
Thank you very much, Tina, and good afternoon and good morning, everyone. I really would like to welcome you to this webinar, which is a follow-up after the publication of the ESAC opinion on GARD. Next slide, please. A few days ago, the EU Reference Laboratory for Alternatives to Animal Testing, EURL-EQUAM, announced that their Scientific Advisory Committee, ESAC, had finalized its GARD opinion. From a SensoGen perspective, we can now state that our groundbreaking GARD technology, which is based on both genomics and machine learning, has been validated by an objective group of international experts. In the opinion, ESAC recommends that OECD includes GARD skin on their list of internationally agreed test methods, which is a major milestone in the regulatory approval process for the GARD technology. This also means that we will be able to offer GARD skin to a significantly broader customer base and paves the way for an accelerated market growth. From now on, we will refer to the positive ESAC opinion in all our external communications. Next slide, please. So, in a brief summary, what did the ESAC working group state? Well, first of all, they concluded that the evidence provided on GARD skin is sufficient and adequate to support its scientific validity. They consider that GARD skin is ready to progress further consideration by the OECD for test guideline development. They also consider that GARD skin is ready for regulatory use. Depending on the regulatory context, positive results may be used as a standalone data source to identify sensitizers. Negative results, however, may not be sufficient standalone evidence to identify non-sensitizers due to limited characterization of applicability domain. Finally, they do not consider GARD potency to be ready for regulatory use due to concerns related to study design, which is leading to limited amount of data supporting predictivity and reproducibility. In his scientific presentation, Henrik Johansson will comment further on these conclusions. Next slide, please. On this slide, I would like to highlight two comments from two of the Joint Research Center scientists that have been involved in the work with GARD. And the Joint Research Center, for your information, is the European Commission's Science and Knowledge Service. These comments can be found in a separate press release that was sent out by the Joint Research Center on July 23. And the name of that press release was "Machine Learning and Omics Arrive in the Field of Regulatory Toxicology." First of all, Silvia Casati, who was a JRC scientist and also working as a group coordinator for this project with GARD, she stated that this is the first time a machine learning algorithm has been independently reviewed for use in the field of regulatory toxicology. Using the code, explanations and data provided by the test developer, SensoGen, ESAC was able to verify the algorithm and reproduce the processing steps to convert raw data into predictions for both GARD skin and GARD potency. So the work that we have contributed to is revolutionary, and that is also why this opinion has taken such a long time to produce. Secondly, Jo Barroso, who is also JRC scientist and an ESAC coordinator, stated that according to ESAC, the GARD skin is ready for further consideration at the OECD for possible development of a test guideline. And these standard OECD test guidelines are internationally recognized test methods used by government agencies, industry, and independent laboratories all over the world to determine the safety of chemicals used in many different sectors, including cosmetics, pesticides, and industrial chemicals. So, two very positive and important statements published by the Joint Research Center. Next slide, please. So, what does the ESAC opinion mean for SensaGen moving forward? Well, already before the ESAC opinion, we have set our direction. With the objective to develop a complete skin toxicology package for regulatory testing, we are gradually increasing our competitiveness both as a test developer and a GLP certified CRO for cell-based toxicology tests. And based on customer feedback, we have already expanded our regulatory test offering with OECD validated skin irritation and corrosion tests that complement our GARD offering. The positive ESAC opinion will drive further customer and partner interest in using GARD skin. And we have actually received some questions about this ahead of the webinar. And I can just answer to those questions and say that yes, in fact, many both potential and already existing customers have been waiting for this ESAC opinion. It either confirms that they have chosen a good test GARD to use, or it confirms that they can start to use the GARD test. So this will definitely help us to develop our sales. Secondly, and finally here, with an OECD GARD skin test guideline in place, we will be able to offer GARD skin for regulatory testing to a significantly broader customer base, especially in cosmetics and chemicals in the EU, the US, and Asia. And to drive this process as quickly as possible, we are in frequent contact with both the OECD and the Swedish Chemicals Agency. This work will actually help us to address a larger part of the addressable market that we have today that is valued at 5 billion. So, with that said, Henrik, I hand over to you for your scientific approach.
Thank you, Axel. And I would also like to take the opportunity to thank all of the attendees for the interest in this presentation. My name is Henrik Johansson, Chief Scientist of SensaGen, and I will do my best to summarize the ESAC opinion from a scientific point of view. But first, I will try to put this into a broader context and just briefly describe the skin sensitization testing in a regulatory context. And the easiest way for me to do that is to start by talking a little bit about biology and immunology. So the mechanisms of skin sensitization are traditionally divided into two phases, known as a sensitization phase and an elicitation phase. In the sensitization phase, which is the first encounter of an exposed individual to a potential sensitizer or a xenobiotic, meaning a foreign compound, the compound is taken up through the skin where it interacts with endogenous proteins. And here they form so-called hapten protein complexes, which are in turn activating both dendritic cells and keratinocytes in the periphery of the skin. Activated dendritic cells then migrate to a local lymph node where they interact with specific T cells of the immune system. And these specific T cells in turn becomes primed both as effector cells of the immune response, but also forming what is referred to as an immunologic memory. So this exposed individual is now termed to be sensitized. And upon any secondary encounter of the same sensitizer, rapid elicitation is being mounted by the immune system and give rise to the clinical symptoms known as allergic contact dermatitis. This knowledge has been summarized in a regulatory framework, which is known as the adverse outcome pathway. And this pathway describes 4 critical key events, which mirrors the mechanistic information of the previous slide. So firstly, we have a molecular initiating event, which is the covalent interaction with cell proteins, which leads to the activation of both dendritic cells and keratinocytes in the periphery of the skin. And this in turn leads to the tissue response, which is the key event 4, where specific T cells become primed and forming the basis of the downstream immunological cascade upon secondary encounters. So allergic contact dermatitis is adverse effect associated with quite large economical burdens, and of course reduced quality of life of exposed or affected individuals. So assessment of skin sensitization is a central aspect for any chemical safety evaluation, and this testing is being performed throughout the product life cycle. So starting already in R&D phases, sensitization is being tested for to prioritize, for example, among candidate ingredients, candidate active ingredients for pharma, or to test for product changes and to monitor the effect of such changes in the R&D pipeline. Moving on to the regulatory phase, testing is being performed in order to register products in the international markets of chemicals, for example, for REACH registration or product safety sheets. In the production sites, a lot of tests are being performed in order to qualify production changes, to monitor different batches, and of course to ensure the safety of the personnel working at the chemical sites. And also in the market, testing is being performed on already finished and marketed products in order to, for example, support marketing claims. I want to highlight here that the regulatory acceptance is primarily of importance for the regulatory phase of the product lifecycle. This is due to the international aspects of this market and the policy that testing should be minimized, particularly when dealing with in vivo experiments. So OECD have a policy which is known as mutual acceptance of data, and this is in order for OECD member countries to accept data that has been produced in another member state. And this is also the reason why test guidelines exist, so that OECD member countries can beforehand agree on what methods should be mutually accepted for registrations. If we have a look at the historical and current assessment of skin sensitization in a regulatory point of view, these are the test guidelines that are currently available. We have traditional testing in animal experiments or in vivo methods, which primarily comprise the classical guinea pig assays from the 60s, the Buehler assay and the guinea pig maximization test, which were in part replaced by the local nuclear assay in the 90s due to its ability to both preserve animal welfare, but also for its ability to provide information of sensitizing potency. In the 2000s, these methods are also increasingly being replaced or complemented with in vitro methods, which are referred to in the test guideline program as guidelines 442C, D, and E. These methods are not sufficient for standalone classification of sensitizers, so they are also combined into what is known as a defined approach, where the data from these methods are integrated to provide a more secure classification. This was very recently published this spring and is available as TG497. These methods can also be mapped to the adverse outcome pathway, which I talked about in a previous slide. So we can see that OECD 442C maps to the key event 1 and then covers the covalent binding to proteins, where we primarily find the DPRA assay and the KDPRA assay, whereas key event 2 is monitored by the keratinosens and lucens assays, and key event 3 are today monitored by the HCLAT, USENS, and the IL8 assay. But we do also see that we have a lot of emerging methods in the test guideline program, where GARD traditionally is being placed under the key event 3. And of course, GARD will now be the main focus of the remainder of the presentation. So this will be an introduction firstly to the technology platform and its applications. But we can state straight away that we consider the GARD to be a technology platform on which several individual methods can be based. So these are today the methods that are commercially available, where the top row is the subject of this presentation and deals with the adverse endpoint of skin sensitization. So we have GARD skin for hazard identification of sensitizing chemicals, GARD potency for subcategorization of identified sensitizers, and GARD skin dose response for a quantitative assessment of sensitizing potency. But I will come back to all of these methods and how they perform and how the ESAC is viewing them. But before that, I would like to just introduce the technology platform a little bit more in detail. So GARD, which stands for Genomic Allergen Rapid Detection, combines the human immune system with genomics and machine learning. And one way to visualize this is to consider the platform as a modular testing strategy framework with 4 technical components that could be customized and interchanged in order to define a complete and frozen method. And these components or modular components would be the biological cell system, the training data set, a genomic biomarker signature specific for the adverse outcome being investigated, and a defined prediction model. And I will go through these different components in turn before I arrive at the GARD skin potency and dose response methods. So the biological cell system of GARD is an in vitro model of dendritic cells. And as I have explained during the early stages of this presentation, these cells are paramount for the decision-making of the immune response, as they direct the downstream activation of specific T cells. And this is being the knowledge of dendritic cells is today quite profound, and one knows that the mechanisms involved in the activation of dendritic cells in response to xenobiotics is quite diverse. The dendritic cells are, for example, equipped with innate immune receptors, able to recognize foreign substances, which are known as xenobiotics. And these receptors in turn activate self-defense mechanisms of the host. This could, for example, include the upregulation of co-stimulatory molecules such as CD86 on the cell surface of dendritic cells. We also have a lot of cellular stress responses. Both oxidative stress are quite common as a damage-associated molecular pattern, as well as metabolic stress due to the metabolisms activated by these xenobiotics. And all of this in turn, of course, then activates the ability of the cells to communicate and, importantly, differentiate how they communicate with other parts of the immune system. And all of these functions in the human cells are, of course, controlled by proteins, and the proteome of the cells are in turn controlled by the transcription of DNA into mRNA. So this is why we believe that gene expression analysis provides a very efficient way to get a holistic view of everything that's going on within this cell type in response to xenobiotic chemicals. So with this knowledge then, and with these assumptions, the basic hypothesis for all development of GARD methods is that there should exist genes and pathways in the dendritic cells that are differentially expressed depending on the stimuli. And if we can identify those genes, they can be used as predictive tools, meaning that they would then behave and form different expressional patterns, whether or not the cells are exposed to a sensitizing agent or a non-sensitizing agent. And the best way to test this hypothesis is to create a training data set consisting of raw data of a reference set of chemicals with known properties of the adverse endpoint that you are investigating. And from this, whole genome data can be gathered and subsequently analyzed in order to find a genomic biomarker signature, which is then a subset of genes that are empirically demonstrated to be associated with the endpoint, meaning that we could identify genes that react differently to, for example, group A and group B, or skin sensitizers and non-sensitizers, for example, in the case of GARD skin. And also one unique feature of GARD is being provided here by the high informational content provided by these biomarker subsets, known as genomic biomarker prediction signatures, as not only can methods, as we saw before, be mapped to the adverse outcome pathway, but we can also actually map the genes and relevant mechanistic events that are being monitored in these biomarker prediction signatures to the different key events. And we see that even though we use a dendritic-based cell line, we do catch a lot of mechanisms that are otherwise being associated primarily with key event 1 and 2. And then finally, the access to a training data set and a defined genomic biomarker signature allows the finalization of a prediction model, which is typically a machine learning algorithm. All of the currently available GARD methods are based on support vector machines. We are also, of course, evaluating others. But these are algorithms that are quite mainstream in biological classification problems, and has been shown to be quite adequate and precise in these types of classification problems. So one of the unique features is also then that being based on this identical strategy framework, really only the biomarker signature and the prediction model is unique to each unique GARD assay. And this comes with certain advantages in the laboratory, as the cellular protocols are identical for all GARD methods. And this means that we can multitask and create several samples from several compounds in the laboratory, and then just divide the readout at the endpoint measurement. So the 6 steps typically used to describe the workflow of all GARD methods then are basically routine cell culture procedures required to maintain the cell line in our laboratory. All test chemicals are analyzed with what we refer to as a GARD input finder, in which we define a suitable input concentration of the test chemical, following which replicated GARD main stimulations can be performed, from which we isolate the RNA of cells and then apply the different biomarker signature required for the readout, which is then the endpoint measurement using a non-string equipment. And having access to the non-string raw data of gene expression patterns, we can then apply the prediction models and our data analysis package to classify the chemicals according to the prediction model's ability. So the presentation today, as I mentioned, will be focusing on skin sensitization. We do have 3 different assays available, and I will touch upon all of them, starting with GARD skin and GARD potency. So the GARD skin method is based on 200 genomic biomarkers, and it has been demonstrated to be quite accurate with a good balance between sensitivity and specificity. It's intended for hazard classification, meaning that it discriminates between sensitizers and non-sensitizers. Contrarily, GARD potency subcategorizes 1A, meaning strong sensitizers according to the globally harmonized system, from 1B, which is then weak sensitizers and non-sensitizers. It's based on 51 genomic biomarkers, and it's also quite accurate in the classification of 1A, meaning strong sensitizers. And the intention is then, of course, to tier these 2 different methods into a sequential testing, also known as the GARD-defined approach, in which in the first tier, GARD skin is applied to identify sensitizing abilities in test chemicals, upon which positive hits can move forward to a second tier, in which GARD potency can be applied to classify strong sensitizers from weak sensitizers. And all of these methods have been evaluated by the ESAC, and this is the data that they had for review. It has also been published and is available as an open source material. The ring trial that was conducted in order to validate the methods was a blinded study comprising 3 independent laboratories and 28 blinded test chemicals. And the figure here shows the 28 compounds that were assayed and the classifications of the 3 different laboratories, which were Sensagen as a lead laboratory, Eurofins in Germany, and Burleson Research Technologies in North Carolina. We can see here that GARD skin is both reproducible and quite accurate, leading to the figures that I earlier reported. GARD potency was then applied to every compound that was identified in the individual laboratories as a sensitizer. And these are the results for the GARD potency ability to subcategorize 1A and 1B chemicals. So the GARD potency was 88% accurate in the accumulated results across the 3 laboratories and 3 independent experiments at each laboratory. The GARD-defined approach then could be calculated to have a predicted performance of 86% accuracy, whereas reproducibility ranged from 60 to about 80% within laboratories and about 70% between laboratories. So the ESAC opinion on these data, as Axel already mentioned, was that both GARD skin and GARD potency, and therefore also the GARD-defined approach, are scientifically valid and fit for purpose, which means that GARD skin and GARD potency, and also therefore GARD-defined approach, may be useful for internal decision making in the industrial setting. Furthermore, GARD skin is considered to be ready for regulatory use, and depending on the regulatory context, results may be used as standalone data source to identify sensitizers, whereas negative results should be considered in a weight of evidence approach. ESAC also concluded that GARD potency is, even though it's scientifically valid, not considered to be ready for regulatory use due to the concerns related to study design, which led to a limited amount of data supporting the predictivity and reproducibility. And so if we were to comment on these conclusions, we do agree that the data made available for review of GARD potency, and therefore also the GARD-defined approach, was in some ways limited. And this was identified by us proactively already last year, where we in fact submitted the complete data matrix for all 28 compounds to the ESAC for consideration during review. However, ESAC has not prioritized to consider this additional data, even though it was specifically asked for in the report. So, of course, we will pursue efforts to understand why that is the case. Simultaneously, we are already in the workings of providing additional evidence supporting GARD potency, both predictivity and reproducibility. And this data is being created so that it will harmonize with existing reference data, which is provided by the OECD defined approach test guideline, which was published this spring, which contains quite potent and standardized references for potency assessment of a large set of chemicals. And we will also produce similar data for GARD skin dose response in order to qualify these both methods for its intended purposes. Finally, as a response to the recommendation to pursue an OECD regulatory acceptance status, we are already in communication with OECD representatives, and the test guideline has been drafted in collaboration with such representatives, so that we are well prepared to pursue the next steps in this process. So I did mention dose response briefly now, which leads me on to the next and last section of this presentation. GARD skin dose response is an adaptation of the GARD skin method that has been created in order to provide quantitative potency assessments. So even though GARD potency is particularly of use for registration purposes, GARD skin dose response has been developed to support decision making in R&D phases primarily, in which the discrete division of 1A and 1B sensitizers may not be enough in order to perform informed decision making in a candidate R&D pipeline. What is desired is, in contrast, a quantitative assessment of sensitizing potency, and we can do this with GARD skin dose response in a full analog to the LLNA, which is the gold standard for potency assessment today. So basically, what we do is we employ the GARD skin prediction model, and we evaluate the response from the prediction model at a range of concentrations, which allows us to identify the minimal concentrations required to elicit a positive response. And this is in a complete analog with the LLNA, whereas the response value of GARD is the decision value, which then is a direct comparison to the stimulation index of the LLNA. We have a binary threshold, which is in the case of the GARD skin, when the decision value progresses from negative to positive, which would then refer to the threefold induction of T cell proliferation in the local lymph node assay. And the readout is then also comparable, where we define the CDV0 value, which would then compare to the EC3 value of the LLNA. And what we find when performing this in a set of compounds, we see that the CDV0 value, as defined by GARD skin dose response, quite well correlates both with the gold standard method, EC3 values from LLNA, but also from human clinical data as provided by the no observed effect level, as can be seen in these graphs. So moving forward, we will pursue and produce additional data for both GARD potency in order to qualify or in response to the ESAC review, and also simultaneously generate these types of GARD skin dose response measurements in order to substantiate the usefulness of such an approach in an R&D environment. And this was the final slide of my presentation, and I would now like to open up the Q&A session of this presentation.
Yes, thank you, Henrik. And this was today's presentations. So thank you to both of you, Axel and Henrik. Great presentations. And we're now ready for the Q&A session. If you would like to submit a question, you already have, some of you, thank you so much. Please do so. As a reminder, do it by using the questions chat box that you can see to your right. And I can see that we have quite a few questions coming in already, which is great. Both Henrik and Axel are here to ask or answer them for you. And we will start with a couple of scientific questions for you, Henrik. What does it mean that GARD skin can be used in a weight of evidence approach? Is this 2 out of 3 in combination with other tests?
So weight of evidence basically mean weight of evidence is an approach that can be taken when registering chemical products. And it basically comprises any knowledge that has been generated about the specific chemical. It can be in vitro data, it can also be in vivo data, it can be clinical human data, if that is available. And also QSARs and read across can be applied or submitted in a weight of evidence approach for registration. So by definition, it will require some sort of expert judgment, which is not the case of a defined approach, where the key word is really that the method or the strategy is defined. So the outcome and the interpretation of the data is defined a priori for a defined approach, whereas it does not necessarily have to be the case for a weight of evidence approach.
Thank you so much for all those details, Henrik. There's another question for you. Please explain the following. Depending on the regulatory context, positive results may be used standalone to identify sensitizers.
Yes, so there are, I mean, the chemical market is an international market, and there are several regulatory contexts available. And what the ESAC means with a regulatory context in this case, I believe, is associated with the different legislations, the different chemical legislations that are in effect, depending on the region of the world you might be in, and also depending on the subdomain of chemicals that you might be operating in. For example, there are different legislations in force when dealing with cosmetics compared to agrochemicals, and in turn, cosmetics may be differently regulated due to legislations in Asia and in Europe, for example. So there are several regulatory contexts in which a test guideline and also the ESAC opinion can be interpreted.
Thank you, Henrik. I will move on with questions related to GARD potency. Is there a chance for GARD potency to get a recommendation in the future? If yes, what is the process for that?
There might be. The process would definitely be that we follow the line that we described in the presentation. We generate more data. We need to come into contact with ESAC and once again consider the additional data that was submitted last year. In fact, all of the data that ESAC requested in the opinion is already has already been made available to them. We need to understand the process of interpreting this additional data as we also generate more data. There's also an opportunity for this to be interpreted differently by experts in the OECD, as there will also be a review by experts in the OECD step of the process. So, of course, having made available all of the additional data to them, the outcome might be different.
It's good. That was also the answer to the next question. The way that GARD potency was tested by ESAC, do you perform the same type of tests? I think that was a question related to if the data was the same that ESAC was reviewing. So let's take the next one on GARD skin. It's related to GARD skin and accuracy. Achieving 94% accuracy in a validation study is one thing. Performing on that level when applying GARD in the real world can be something else. Do you expect that GARD will have an accuracy around 94% going forward when doing predictions for customers?
That's quite right. The accuracy will, more than anything else, depend on the chemical domain, the chemical subset domain in chemical space in which the method is being applied. As ESAC also mentions in the report, the applicability domain and the limits of the applicability domain has not been fully explored, which is the case for all of the in vitro methods and also the local lymph assay actually. So that is something that we are quite well aware of, and moving forward this will be monitored and collected as evidence for different applicability domains.
Thank you so much, Henrik. I will move on to the next question. ESAC mentioned a reduced prediction signature based on 10 genes. Will you change to that, or will you stay with 200 markers? What consequences do you expect from replacing your algorithm or staying with a high number of markers with ESAC suggests do not contain signal?
Yeah, this is quite known to us, and it's not at all a surprise or any news. ESAC is quite correct that the number of biomarkers could be reduced without large loss in information or predicted performance. The reason that we use the prediction signature of 200 genes is primarily from a historic context. So these biomarkers were empirically identified from a data-driven approach from a whole genome dataset, where the 200 genes were being identified as optimal. Upon the tech transfer to the NanoString platform, we quite intentionally used all of the 200 genes in order to demonstrate that the platform change would not have an effect. Had we made any major changes in the biomarker signature at that point, the effect of demonstrating the usefulness of the technology platform would have been lost. And moving more forward from there, our R&D team has identified several different possibilities to reduce the signature, but we have opted not to do so for the regulatory application, at least, just because we do not want to mess around with anything. Because once we start changing any parameters specifically for the biomarkers that we evaluate, all of the data that we have historically generated would no longer be supporting evidence for the function of the method. And it's also a way to build credibility during time that we do not change in the classification algorithm and the features that the classification algorithm is based on. So I would say those are the reasons primarily that we stick with the 200 genes. And then I'm quite aware of that there is a discussion of overfitting, and I think it's of crucial importance to consider overfitting in every type of predictive modeling. GARD skin is no exception. And this is also why we do monitor overfitting quite closely in all our R&D activities. During method development, we follow overfitting with cross-validation exercises. But most importantly, of course, it's being controlled by the evaluation of independent test sets, which is precisely what the validation is all about. So even though the risk of overfitting is real, I think the data present demonstrates that GARD is not overfitted.
Thank you, Henrik, for that great explanation. I'm going to go back and do another technical question. Why is the reproducibility not the same between the different laboratories?
I'm sorry, I did not quite catch the question.
Okay, the question is the reproducibility is not the same between the different laboratories.
You mean within reproducibility then?
Maybe we need to interpret the question. Why is the reproducibility not the same between the different laboratories?
Oh, I mean, in general, I would like to comment the question by stating that all reproducibility that is not 100% is due to variance, right? And the variance may have different impacts at different times. That's in the nature of variance, and the lower the variance, the greater the reproducibility. And these are the figures at hand.
Okay, thank you. Perfect. I think that's a good answer. Before I let Axel in for a question, I'd like to finish off here with a question related to GARD air, the test for respiratory sensitization. Is GARD air affected at all from this ESAC recommendation?
No, not directly, but I would like to believe that the approval of GARD skin will make it easier for any method and any method developer to submit and get methods approved based on these types of technical components that we use, meaning machine learning algorithms, high informational content data, and so on, and also cloud-based softwares for analysis. All of these technical components are also shared by GARD air, and one of the reasons that this opinion has taken quite a lot of time is for OECD already at this level to review those technical components. So those obstacles will now have been removed if and when we move forward with a GARD air submission.
Thank you so much. I have only one question left on the list today. Maybe if you want to submit a question, please do so now. Use the questions chat box, or I will do the last one now. It's for you, Axel. Are you ready to give estimates of this year's sales, or next year, or in 5 years?
No, I'm not. I mean, we will release our half-year report in August. But I can repeat what I said in the beginning of the presentation today, and that is that the fact that we now have a positive ESAC opinion for GARD skin is actually extremely good for us at SensaGen, since we have received that question quite a lot from both customers that we already work with and also potential customers. And the ESAC opinion now confirms that this model really works. You know, we knew it already, but now it has been also noted by an objective peer group, which is for us key. And that also means that since, for example, GARD Skin has been used in R&D projects, now the companies that have used that know that the test is validated, which will help, I guess, more R&D teams to use GARD, which is good. And then, of course, another thing that is important here is that the feedback we have received from our different partners is that when you receive a positive ESAC opinion, it is much easier for us to start to market GARD Skin towards our customers and for us to set up GARD Skin in our laboratories. So I see this as very positive, but I don't want to give any estimates when it comes to numbers.
Thank you, Axel. That's perfect. There is a follow-up question on this as well. When will you make penetration rate goals for the TAM, for the TAM? Any comments on that, Axel?
Sorry, I didn't understand that question.
When will you make penetration rate goals for the TAM? Maybe we need to ask the person to clarify the question a little bit. Let's see what happens, if we can get a clarification here so we can see if we can comment and get a reply. While doing so, the GARD Air discussion also triggered another question. Will there be any studies for peer review of GARD Air? Any peer reviews in the plans for GARD Air? Henrik, do you want to comment on this?
Not in the immediate future. We have generated quite a lot of GARD Air data, primarily in collaboration with industrial partners. And when doing so, it's always important to be in to be in sync with the partners in order to publish anything that you do. So GARD Air is being used in R&D phases by certain market segments, and once we are ready to publish anything, of course that will become a priority, but not in the immediate future as I see it right now.
Okay, thank you. And we did get the clarification of the question as well. The TAM is short for total approachable market. So the question is, when will you make penetration rate goals for the total approachable market?
Okay.
Is that in your
plans? Yeah, then I understand. No, I mean, we have already stated that our addressable market is approximately 5 billion SEK. And I mean, what I also said in the beginning of the presentation is that our opportunity to now take more of that market faster definitely increases with the ESAC opinion on GARD Skin in place, and hopefully also the OECD adoption of GARD Skin that we hope shall follow. Apart from that, I think that the most important for us is still to work towards our break-even goal and to show that we can have a steady revenue stream. Our break-even target for the end of 2022 still stands.
Perfect. Thank you, Axel. This was the last question of today's webcast. So that means that we have reached the end of this webcast. Thank you for participating. Thank you to our speakers, CEO Axel Sjöblad and Dr. Henrik Johansson. And as mentioned before, the recorded version of this webcast will be posted on our web pages shortly. You will find it on the investor pages, but also on the landing page. So we just want to say thank you very much and have a great day. Bye, everyone.
Thank you very much. Goodbye.
Thank you. Bye.